just have one slide, and I hope everyone can see it. So my name is Jason Williams. I'm coming to you from Cold Spring Harbor Laboratory, New York. And I'd like to welcome everybody to this first webinar of the NIAID-funded Bioinformatics Resource Centers for Infectious Diseases. So currently, this is a group of 3 different distinct projects, which all have the same mission of providing tools and computational platforms that are really going to support infectious disease research and all of the people in those communities. Of those 3, we have the BBBRC, BRC Analytics, and the Pathogen Data Network. And this, as I've said earlier, is our very first webinar where we're trying to get all of those groups together. Initially, we hope to present this the second Friday of the month around this time to help grow an audience and to get a chance for both the researchers and these different platforms to know what resources are available. In this initial webinar, we thought we would give a little bit of an introduction to each of the different platforms so people can begin to know what resources are available or will be available at each one of them. And in the future webinars, we are going to focus them on individual tools or workflows. So with the hope that in a half an hour of joining each one of these, people would get a chance to see some capabilities, ask some questions, and also tell us a little bit more about what you'd like to see and learn more about. So with that said, we are going to have approximately 8 or so minutes from each of the different platforms to give us a chance to get to know what resources are available. And I believe first we're going to hear from the Pathogen Data Network. And let's see, Aitana, will that be you, or do we have Christoph as well here?
Yes, it seems it will be me.
Okay, I'll stop sharing and I'll give it over to you.
Sounds good. All right. So welcome, everyone. It's my pleasure to present the Pathogen Data Network. Our motto is a world empowered by data to tackle infectious diseases. The mission of PDN is basically to enable a worldwide ecosystem of linked data and tools as well, because it all comes together to support both research and public health response to infectious diseases and outbreaks. We have four main missions. First, to foster data discovery, because that's the first step. You need to find the data in order to be able to do something with it. And that's through the integration, referencing, and annotation of scattered data and resources into single portals. And we'll see a bit more what that means, but you can think of that like the central pathogens portal is a resource from where you can go and find data. To also support efficient surveillance and outbreak response through innovative computational approaches on pathogens of interest. So this means developing new resources, a new bioinformatics pipeline for particular pathogen, and also trying to make the best out of novel technologies, notably AI-based. To also enable timely data sharing through the development of private data sharing environment, as well as also looking into all the aspects around that, all potential barriers to timely data sharings that might be also linked to ethical aspects or policy aspects. And last but not least, also preparing the next generation workflows through training and capacity building. So what we achieved so far, you can see on our website, we have a bit of an achievements page for this first year, but we notably developed further the infrastructure. So notably the EMBL hosted pathogens portal, with notably also a page dedicated in particular to finding wastewater data to support faster outbreak response around that data type that is used a lot in many countries now to support monitoring of pathogens. And we also work towards establishing a global network of stakeholders, and notably this vision of having a global network of scattered pathogen portal nodes that this is something that we could start with already pathogen portal nodes that have been established in Central and Latin America, in Africa, and also in Asia. And so basically PDN keeps building on this success. We continue with the same mission, but there are a few twists due to new NIH policy, so we need to change a few things. So we had notably to update the consortium because we were no longer, I mean, we foreign subawards were no longer awarded. And nevertheless, we could find a way to keep the same core mission unchanged. So that's very important for everyone to feel reassured. And we will basically build on the first year network to also ensure adoption and sustainability. But basically, the tools that will be now further developed will have a little shift on tools that are developed within the SIB network. And we continue to work with the US partners at Cold Spring Harbor and Georgetown University. So you can see here several tools you might have heard or used, some or several of those. You might maybe not know that all of those have in common that they are developed or co-developed for some of them by the Swiss Institute of Bioinformatics. So just to try to explain a bit more, what we will be trying to do is to really focus now on actionable insights. So to give practitioners a clear, up-to-date view of outbreaks. So basically through the tool GenSpectrum, which is a platform that allows really to, for specific viruses for now, but we will be extending that to additional pathogens, bacteria, fungi, is to be able to really follow specific variants and mutations of interest and see how they evolve over time and how prevalent they are also in different places of the world. This platform, GenSpectrum, is really a portal from where you can look at this data, but it is powered actually in the backend by a technology called Loculus. And Loculus is actually a reference software that is available also open source and then can basically power pathogen data hubs. And so Loculus itself will be extended, and it will be fed with public data from the NSDC that then will be searchable through GenSpectrum. And if there is any data that is submitted also directly to Loculus, it will be also shared to the NSDC. There is also still a focus on wastewater data and outbreak detection with that data type. And we will in particular integrate in Loculus various additional analysis. And currently Loculus supports viral analysis. It will be extended with some tools that are used in Switzerland for the Swiss wastewater surveillance program to then basically then annotate all the public wastewater data. So this is called VPIPE. Regarding the focus on developing innovative technologies to understand better pathogens, we will focus principally in using AI tools to accelerate the understanding of emerging or unknown pathogens. So here there are two aspects. One of them, actually more to the right here, is to take advantage of current AI-based models, notably to predict protein structure in 3D, to annotate viral proteins that are unknown thanks to this protein structure homology somehow. And this is something that has already started a bit within SIB through the work of notably Philippe Lemercier, who is the person behind ViralZone. For those of you who know that resource, I think that those working in viruses generally all know ViralZone. And so since there are many proteins, in particular in the viral realm, that are completely non-annotated and unknown, this will be a useful integration into existing knowledge bases. There's also the idea to advance tooling, in particular regarding the computation of trees, both for viruses with what currently expanding NextRain, also for bacteria, expanding tools like BEAST, that some of you might know as well, and also using, again, these novel developments in AI to be able to build also trees that are based on structural homology rather than sequence homology. And this is also something that will be then developed further within the new consortium, and also the pre-computed trees will also be made available. And another important aspect here is to foster fair data sharing from around the globe. So I mentioned before Loculus, which is basically a reference code that can be used to deploy a pathogen data hub. And today, Loculus is the backend behind GeneSpectrum that I have mentioned, but also is actually the backend behind Pathoplexus that many of you might know as well. And so by expanding Loculus, by expanding the capabilities of Loculus to annotate also not just viruses, but also bacteria, fungi, and currently Loculus can only start from assemblies, but also now the aim is to expand it to also raw data sets and also wastewater data sets. It means that ultimately, if Pathoplexus, for example, downstream would be interested in having also those data types, it could implement those because the backend would already be able to accommodate for those. So it's really also making sure that the hidden tools are expanded so that then they can be further used by those who rely on them. And also this same technology could be used for people around the globe to set up their own local data hub, which could then also foster fair data sharing. So, and that's just last just to say that we continue on the ethics training and outreach. So please connect with us, and thank you very much.
Okay, next we'll hear from the partners at the BVBRC. So I think Rebecca and Anna will take care of that for us.
Yeah. So we're at the BVBRC. My name is Anna Kapir. I work as an outreach coordinator and bioinformatic analyst at the J. Venture Institute. Rebecca, if you want to introduce yourself really quickly. Rebecca, you're on mute.
Sorry, I was muted. I'm Rebecca Wadham from the University of Virginia, and I handle bacteria outreach.
So on the next slide, you'll see the key features of the BVBRC. And so we support diverse bacterial and viral communities. The BVBRC is a resource that has existed for years at this point, so continuing to build on that. But we also annotate and manually curate metadata as the data comes into the resource, and Rebecca will tell you a little bit more about how much data we have. We use explainable AI and machine learning based tools to add value to the data as it comes in, similarly with our unified data model real-time integration processes. So we're taking data from publicly available resources and making it more useful for you to do your analysis. Then once you have all of that data at your hands, we have these modular, interoperable, high throughput analysis services that will allow you to analyze that. And then you can also analyze your own private data in your workspace. And we'll tell you more about all of this in subsequent webinars in this meeting, but just wanted to give you this high-level overview of what is housed within the BVBRC. But it is set up to support exponential growth of pathogen data. So a few years ago, we didn't have any SARS-CoV-2 genomes, and now we have millions upon millions of them. And we continue to analyze multi-omic data using visual analytical tools. And if you're someone that uses high compute resources, we have programmatic and batch access via APIs, the command line interface, and FTP. as data comes into the BBBRC, it's consistently and accurately annotated, and we derive other data types. We have a focus on outbreak tracking and phylogenomic and epidemiological analysis for rapid outbreak responses. We continue to offer outreach and education like this webinar right now. And so you might be wondering if you can see yourself as a BBBRC user. So on the next slide, you can see who our BBBRC users are. We have over 70,000 registered users that come to the site for 25,000 times a month. We have over 20,000 citations, and our community continues to grow. These users are people from basic infectious disease researchers and biologists across academia, government labs, nonprofit, industry, and pharma. We also have clinical researchers, epidemiologists, bioinformaticians, data sciences, and people that provide other data resources as well. And I think Rebecca will tell you a little bit more about our data on the next slide.
We bring in data every night from GenBank, and while we keep the original annotations, we also re-annotate, especially on the bacterial side, so they have consistency and one-to-one comparisons. I've been with the BRC since our personal BRC started with 8 bacterial genomes, and we now have 1.2 million. So as the data grows, you can see that the viruses swamp everything, which forces us to look at log total genomes. As that grows, the complications are, how do users find the data that is of interest to them that will help them analyze their own private data? And so when we bring that data in, we also bring in the metadata from GenBank, from BioProject, from BioSample, and we run crawlers on that to sort out the metadata to provide users with filters so that they can quickly and easily determine something as simple as how many Staphylococcus genomes were isolated from blood in Egypt. And you can do that almost immediately, and that takes this huge tarball of data and makes it accessible to our user community. So it's not only about the data, it's also about analyzing it. And right now we provide 33 different services to analyze the data in BVBRC. Now what comes with this, if you submit some reads for, let's say, an assembly, you get your assembly in contigs, but we also try to provide informative reports to give you indications about the quality of your data. And also, we're all primates. We see things that resonate with us. These are just a few of the viewers that we have to help people see their data and understand it. And I should point out that many of these viewers are in the publications that we see that our users are doing, because we also provide them as scaled vector graphs so that people can take them directly to publications, which is what we want. We want them to move forward their research and be successful and cite us. That's always important. So who are using these tools? Of those 70,000 users, are they really using those tools? We've been keeping track since we started providing bioinformatics services in 2015. And over the years, we can see that there's a lot of use of the tools. What I've broken it down into is the top 10 tools in the last year, and 2025 just goes through August. So I expect it'll match 2023. 2021 was a year we were hit with the bacteriophage community wanting to do a boatload of phage annotations because we just launched the pipeline. So you can see that's why we have that huge spike there. But always, assembly and annotation are the primary drivers that people like to use and use all the time. BLAST, of course, one of our the good things about our BLAST service is you can blast it against private data, against specific taxa. So that's and the users can collect groups of data and then blast against that to see if they have, say, the antimicrobial resistant genes of interest that they're looking at. And also our trees are very popular as well. And more we're seeing more metagenomic analysis creeping in. We have taxonomic classification and metagenomic binning, things like this that we keep expanding on to provide our users with the things that they're most interested in. So when you're providing bioinformatics services, you have to reach out to people to show them how to use those services. So we try to do everything. We have documentation sites. We have quick start guides. We have tutorials. We have instructional videos. We also have a bioinformatics course called Bacterial Bioinformatics on Coursera, which has now more than 20,000 people signed up for it. And we're launching 2 new courses in this next year, one on analyzing antimicrobial resistance data and viruses. We're going to do 2 new courses on that. So I encourage you to look at our YouTube channel, to look at the videos that we have there and explore the tutorials.
On the next slide, you'll see a spotlight for our AI Copilot. So on the BVBRC in the bottom right-hand corner, you can try that out to start answering some of your questions. It knows all about the BVBRC and our documentation to help you analyze anything you might be interested in. And on the next slide, you'll see about our next session. So next month, we'll be talking about our outbreak services. So the BVBRC will be leading the next outbreak webinar. Not outbreak webinar, sorry, Tools for Tomorrow webinar. And so you'll hear about our core genome MLST service and our whole genome SNP analysis. And you can see some visualizations of that on this slide. If you
I don't know where those are.
If you
I
hit the next slide button. Oh, I guess they're gone. But this is the team that brings you the BVBRC, and we are always here to help and happy to have you as users. So please reach out if you need anything. We'll hand it over to our friends at BRC Analytics.
Okay, welcome.
All right, thank you.
Anthony.
Thank you. Let's see if I can. All right, I have only one slide. So what BRC Analytics is, it's an environment which allows you to do essentially entire analysis from data to publication in a browser. So it connects to 3 main sources of data in CBI datasets. This is our source of truth for reference genomes. To UCSC BRC Hub. This is the source for annotations, because it also allows you to incorporate community annotations. And it connects to full SRA sequence read archive using ENA API. It pulls analysis workflows from IWC Consortium. It's a Galaxy thing which stores community curated workflows. And all of this is happening within the Access CI public cloud infrastructure in the US. So it has virtually unlimited compute resources, and our head nodes are at Texas Advanced Computing Center. So that's my last slide. I'm just going to show you how to use it. So our point is to let people actually do analysis. And let me see. So let's suppose you want to do some, I don't know, viral surveillance analysis. I chose for this particular demonstration, I chose measles just because it's small, but you can do it on anything. Maybe you want to do surveillance of latest yeast pathogens or malaria or something like this. So let's start with measles. So I'll go find measles. So for measles, I have one assembly. That's the assembly. So BRC Analytics provides you an opportunity to look assembly in the browser, go to NCBI datasets, do taxonomy, look taxonomy, all these things, but that's not the killer feature here. The killer feature here is this analyze button. So again, what I'm trying to show you here is a surveillance example. So in a surveillance example, you have samples from multiple individuals across geographic locations, times, whatever. This is just one of the examples. For example, if you want to do RNA-seq for yeast, or ChIP-seq or ATAC-seq analysis on Plasmodium, you can also do this here. But again, let's stick to malaria and let's stick to surveillance, and surveillance is usually variant calling. So I'm going to go pick a workflow. There are two of them, but which are kind of geared towards this virus. Remember, this is an RNA virus. And once I pick the workflow, I can configure inputs. So when you do surveillance studies, what you essentially do, you collect data from individuals, you map it against the reference, and you call variants. So the reference we already picked. We need to know gene information because essentially we're going to classify all variants, whether they're in certain genes that are recording or non-coding. So you need gene annotations from that. That is what pulled from UCSC. And finally, you need the data, the surveillance data. This could be your data, so if you do surveillance in a hospital or things like this, or it immediately queries SRA, so it already knows that there are, for example, 446 data sets for measles. Not that much. It's not COVID. This is one of the reasons I picked it. So I'm not going to analyze them all. I'm just going to probably pick the biggest ones. So let's pick this one, that one, that one. And well, that's probably good enough. So that's 225 samples. So I just click add 225 samples, and I click this launch in Galaxy button, which would bring me to something which looks like this. So this is already a launch interface. You can see that my samples are already here, so they would be automatically pulled from SRA. My reference is here, my gene annotations is here, so I just click run workflow. And once I do that, I get to this page where you can actually see the workflow invocation. That's the workflow that is being run. Of course, I pre-ran it. I'm not going to do it in 5 minutes. And these are my results. So when you do variant calling, your results usually, at least on the surface, they're pretty boring. They look like this. But again, it's Galaxy. So right now in Galaxy has unlimited number of visualizations. And what we can do here is something like, for example, let's load this up into Jupyter Light instance. And so the cool thing about Jupyter Light instance is that, in fact, this was sort of a co-edited together with LLM. So I generated this code with LLMs. I even have prompts there. But it's linked to this data, so I can see, okay, these are the variants. If you know anything about measles, you know it has these weird overlaps. So this is interesting how substitutions are in these overlaps because they would affect overlapping reading frames differently. So I can look at that. I can do analysis of this notebook, restrict things to certain sequences, move them back into Galaxy. And then I can run more Galaxy tools. For example, I can run evolutionary tools such as, okay, which of these sites are under selection? Where is measles moving? Are there any, sites which aren't there, for example, diversifying selection? So I can extract these things, put them back into my Galaxy history. And again, as I just mentioned, I run meme on them, for example. This is the analysis of selection analysis on one of those data sets. And so this would look like this. And well, of course, you need to know a little bit about what this is. But basically, you're looking for, in this case, lowest points. So here are two sites which are likely under diversifying selection. So I can look further what they are, whether they're in overlaps and so on. So pretty much it's exactly 4:30. So in about 5 minutes, I took you from the idea, I want to do surveillance on the virus, through pulling all the data, running analysis. Of course, I pre-ran them. They wouldn't be done in 5 minutes. The whole thing that I'm showing you would take you probably about 2-3 hours to run to the end. And you can take these images and just publish them. So that's it.
All right, what a wonderful way to end, because essentially you'll be seeing more of that from this group in the future, a chance to come together. As you saw earlier, Baby BRC will be the next to present. We'll have time to go in depth on specific tools and workflows. So we're at the end of the half hour, but I would say that anybody that is staying on for another minute or two, if there's some questions and people want to raise their hands, we can take those questions. But ultimately, we'll want to be hearing from you as to what are specific features and tools that you need help with. And then each of the partners will respond to be able to make sure that what we present at this call is responsive to those needs and gives you options you knew about or maybe didn't know about to analyze your data. So with that said, I'll end it here for the recording, and I think the presenters will stay on for another minute or two. If there are questions, we'd say to raise your hand or use the chat. And thank you for joining us.